Good evening, everyone, for who's already joining. We're going to wait a few more minutes to let people come into this webinar, but welcome to the webinar to present the results of the Neretva Science Week. Okay, great. Again, welcome everyone to the webinar that is about the results of the Neretva Science Week, where we will be discussing the results, the impact, and also the outlook to the next Science Week. Thank you all for joining. And I will welcome throughout this webinar the different scientists that joined us and the various NGOs that helped out. So yeah, join us on this journey to the Neretva River. As most of you know, this Science Week took part as a part of the Scientists for Balkan Rivers network, which is a network that officially kicked off about a year ago in January, and which was inspired by the work that was already done on the Vjosa, where several Science Weeks took place before. And this year we took it to the Neretva River in Bosnia and Herzegovina. Today I will be taking you first to the Neretva River and the threats that it's facing, and then we'll have time to actually dive into the results with the various scientists that joined us. Later we will be looking at the impact of the Neretva Science Week, after which we have time to actually look out to the Neretva Science Week in 2023, which I'm already happy to announce. It's going to take place from May 30th to June 7th. The science will already be welcome from the 28th onwards, as that's a weekend, so we'll have camps set up for everyone. But yeah, we're excited to dive deeper into the science of this area, of the beautiful Neretva. Later in this presentation, we will be diving a lot more into the whole concept of this week, so please hang around until that moment. But already get excited about the dates and make sure you put them in your calendars. In the next few photo series, I'm going to take you to this to the Neretva River, which is in the south of Bosnia and Herzegovina. And as you can see, it's within this beautiful elder forest. It's very untouched area. And if you go up, and one of the first tributaries of this Neretva River is this Krupac River, which is a little karst spring river that comes into the Neretva. And you see that we climbed all the way up to the very source of this river. At some point it creates some beautiful waterfalls, and then contributing to the Neretva River that is slowly growing over different riffles, forming a lot of pools as well. And then a bit further down, you see that suddenly the water spreads through the whole, through a very big floodplain forest. So it really gets a bit more space to create several ecosystems. And then only a few kilometers afterwards, you'll see that it actually starts cascading a bit more. So you see that constantly the ecosystem changes, which makes this river pretty unique up there. And with all this changing ecosystems, it's really actually part of a way bigger ecosystem, a very untouched elder forest, which I think all of the scientists agree that that's a big reason why the river was so biodiverse. And slowly winding its way down, you see that the river grows and suddenly gets a lot more space near Glavaticevo, the area where we were, to then again be restricted by the geology and wind its way through a steep tight canyon that is characterized by all the little karst springs that come into the canyon constantly. So as you can see, a beautiful section of river, but we wouldn't be here if it wasn't for this river also being threatened by the dam flood that you see in the whole of the Balkans. I'm sure all of you know this map and are familiar with it, where all the red dots mean a dam that is currently being planned, and all the black dots are dams that already are completed and operating. And as we were at Neretva, also we encountered the current construction works of the Ulok Dam, which is very near the research sites where we went. So it's a very vivid problem in this area, which is the reason why we went there to really show the value of this river and to hold this kind of construction of dams. So going back to this map, I'll show you a bit where we were. Like you see this area, which is basically the side of Bosnia and Herzegovina, just south of Sarajevo. It's where the Neretva River flows. You see it bending from where it starts, the mountains, goes all the way northwest and then bends its way down and then makes its way to the Adriatic Sea. And as you can see in the middle, there's a few black big dots already. The middle section is quite characterized by those dams and several reservoirs. But if we then look up to this upper reach of the Neretva, you see a lot of red dots, but nevertheless, none of them are finished yet. And all of this section is highly untouched and still flowing free. As we can see from the value from all the scientists that have been going there, that it's a really unique area. So zooming into this area, which was the area of the Neretva Science Week, we defined several research sites, and we basically took all the scientists that wanted to join us to all those different places and allowed them to dive into the biodiversity and into the functioning of this whole river. Here in the middle, this gray dot, this is Ulok. So this is just upstream from this currently being constructed dam. And this was the first base camp of the camp. And I'll take you there in a few photos. You see this little hut, which is at this moment the most active place of the town, I would say. Years ago, it was a vibrant town with thousands of inhabitants, but at this moment, like maybe 10 are left. But this little bar was really the heart of it at the moment, and it's right next to the river. And it was the place where we set up camp. Some of the scientists set up their temporary labs as well. We camped under the stars, beautiful evenings, and the biggest light that you would see was actually the light traps that scientists set up to attract all different kinds of insects. And it was kind of the highlight when walking back to your tent, you would be diving into all the different insects and the moths, and really impressive amount of insects that were attracted by those light traps. During the day, different scientists, like they all went out to the river looking for the smallest little creatures that live in and outside of the river, looking into mollusks here, finding several really special species. Some people look more into the macroinvertebrates, the insect living down and just flying over the river once they're hatched. Creatures like this one. Then we had a fishing team who in the upper reaches did some fly fishing, and lower down they had some amazing results while snorkeling. We'll get back to those later. And then we had different teams that actually looked more at the functioning and the functioning of the ecosystem, and did some more physical research. Some of them actually taking a boat out as well to reach the least accessible places of this river. In the evenings, we had some moments to actually connect as well. We had a chef cook, Levente, who cooked the most amazing chile con carne and sin carne. We had a band, we had a beautiful concert in the evening, and we were accompanied by a lot of journalists as well, who went out to take amazing shots. Not only that, also managed to get the word out in different newspapers and articles. So we're really happy that all of those different things came together during this week, and it was at the same time as a moment for defending the river, it was a moment to celebrate this river. Now I'm going to give the word to Gabriel Singer, who was the lead scientist of the whole week, and who's going to introduce all the other scientists that are going to lead us through the results of the Science Week. Gabriel, up to you.
Hello, everybody. It's my pleasure to introduce you, and Vera usually introduces me as the lead scientist, but I was not the only one. Whenever I made a mistake, I tried to blame 3 other fellows who tried to plan this thing with me. Predrag Simonovic was among us, Sodin Merdan, and also Stephen Weiss. Vera, can I have the next slide? Maybe, yes. So what is this Science Week? No? And I'm asked as a scientist to introduce this. And I think the scientists that you can meet on such a Science Week are simply engaged scientists. I think of them as such. Our aim is basically to contribute to protecting rivers. And we want to do this as experts. So we feel a bit on a mission as such. And our aim and the way to do this would be to collect data and then increase visibility for what we think is a rather unique river system at the scale of Europe. And the approach to do this is basically to concentrate our expertise in a good, inspiring atmosphere. And I think the pictures from Vera actually showed that that's already a pretty good setup, right? Next slide, please. So we rounded up about 50 experts from across Europe to basically study what we call taxonomic as well as functional biodiversity. Taxonomic biodiversity means that a lot of us were experts on very specific organism groups, like water beetles, for instance, or specific other orders of insects, for instance. But some of us also looked into ecosystem functioning and what we think of as, I would say, functional biodiversity in that sense. We wanted to collect data on primarily aquatic biodiversity, but also terrestrial biodiversity, because rivers have an influence on their surrounding, and they're also very tightly connected to this. And the aim was to potentially assess an impact that may happen by hydropower development. This is a maybe, because we hope that some of these things are never built, or most of them. And we wanted to do this mostly with regard to threatened habitat types and with protected species. The usual approach to do this is very local. So you go to a place, you try to assess what it's like, you try to find species at this place. But we wanted to actually achieve this on a regional scale by distributing sites along a relatively long stretch of the river. And Vera has already shown you the sampling sites to actually achieve an assessment of biodiversity and ecological integrity at the regional scale of the river network of the Upper Neretva. And simply because it's a pretty remote and wild place, in many aspects also dangerous, this necessarily meant sort of an expedition approach to which many of us actually looked forward. Next slide, please. So where did we go? This was the habitat for us scientists for this Science Week. And the graph actually shows the length continuum of elevation, so you can read the slope a little bit. And the part that's circled or in this red square here is basically the region where we were, right? So downstream of this, you can see there is already the Jablanica reservoir, and then there's a chain of other reservoirs until Mostar. And it's the red box that actually shows where we were, and you can see in the pictures above how the river actually looks like. And the third diagram on top actually gives you an idea of the flow regime of this place. So it's pretty seasonal. We have a low flow in the later summer season, but we were there at a time when actually there's a reasonable amount of water flowing. This place, this Upper Neretva, is an incredibly diverse landscape. It has very heterogeneous habitats. And I tried to show this with a graph of a specific parameter that's here called K600, which is a descriptor of gas exchange velocity. And that actually sounds very technical, but you can think of every dot as being on this map as part of the river network. And the color is a very important descriptor for ecosystem functioning. It's the amount or the magnitude at which the water can exchange gas with the atmosphere. And from this, you can actually read the heterogeneity or the habitat diversity of this place. The two lower figures actually give you an idea of what happens to a river when you dam it. This is the flow regime from 1939 compared to 2019 at the most downstream place after the Mostar Reservoir. And you can see the river is pretty dead in terms of its natural flow variability. Next slide, please. So this was the place for, as I said, about 50 different scientists, and there are a few whose results you will see on the upcoming slides, and there's a few whose results we could not include. I would like to point out a few of them. For instance, Emir Delic actually explained to us the local botanical diversity. There's an incredible diversity of forests associated to the river, but also to the slopes of the valleys. Manuela Habe was the one who found the links for us, or at least say traces of it, right? Sodine was part of Sodine Medan was part of the science lead committee, but also looked into reptiles into place, and Sarah Höfler, who's also here today, who actually produced the slide that I've just shown before. Thanks to all that were here. And now let's dive into some of the results that these people actually created. And I'm passing on to Michaela.
Thank you, Gabriel. I have now the honor to guide you through some slides that will present you the biodiversity that was found especially in the insect orders that were gathered last year at the upper stretch of the Neretva. First, we will start with the aquatic insects, with the groups of Ephemeroptera, Plecoptera, Trichoptera, and the superfamily of Diploidea from the Diptera. In total, there were 103 species found there, and the amazing number of 82 species that are classified as rheobiontic species. And these are species that are especially vulnerable to dams, as they need a very high current of the water flow. And then there were found 32 Balkan endemics, and also these species have a rather high conservation status, as they only have live in a restricted area. Yes, and then there was only also one species even new to science. I will show you later on. But the results don't only show you that there was a big biodiversity found there. It was also extremely high in biomass, and there at the light traps that Vera already showed before, they were up to 40,000 specimens caught per night. The majority of these Trichoptera that you can see here on crawling on the stone in the water, they are rheobiontic grazers from the genus Agapetus, and here on the right side you can see these black dots. These are the corresponding adults of this genus. And here I can proudly present, I didn't collect it, but it is Beura neretvaensis. As you can see, it has the epithet of Neretva, and it will be described very soon in the Journal of Zoo Keys. This species was already found in Slovenia and in Montenegro, but not in that very high numbers as we found it at Neretva. Additional several Empididae were found new for Bosnia and Herzegovina, and these results will also be very soon published at Natura Croatica. And additional two species, one species from the genus Isoperla and one from Perla, they have still an unclear status, but they're under further investigation, and perhaps there's some more new that we have found there. Now we'll show you the results of the aquatic Coleoptera. It's aquatic Coleoptera sensulato, because I didn't only collected the beetles directly in the water. I also wanted to concentrate myself on the river's edge. And there were 57 different species found out of 11 families, with 7 Balkan endemics and 21 rheobiontics, and 7 species are also new for Bosnia and Herzegovina. And there were not only Balkan endemics to find. Here you can see Hydrina bosnica. It's an endemic of Bosnia-Herzegovina. It was described many years ago, and the type locality of the species is located in the Neretva Valley, and this is the only region where this species can be found. There is also one family that is new for Bosnia-Herzegovina. It's still not listed in the catalogs. It's a very small species. It's only 1.2 millimeters in size. It has a very cryptic lifestyle and is found at the edges, muddy or sandy edges of the rivers, which have especially natural flow and with very unpolluted water. And here is another beetle found, is a rheobiontic species, also rarely found, but here at this location, you can see here they were quite very rich in individuals. It's an Elmis puberula, and they live in an interstitial at fast-flowing waters of coarse gravel. Now we come to the more terrestrial groups. Because also the floodplains are interesting, what is to be found here, and there were specialists collecting carabida, so-called ground beetles, and there were found 57 different species, and they collected at different localities and divided them into different stretches. And as you can see here, they were different in species numbers, but they were also different in the species assemblages. And at 3 sites that were examined, 70% ratio was found of riparian floodplain specialists, which is a very high number. And here there are 2 representatives of these beetles. This is Bembidion conforme and Bembidion monticular. And they are normally more common in Central Europe and very rare in the Balkans. But they were also found rather in high densities at the floodplains. And this is a good sign for these habitats. Now we come to the Lepidopteran results, the butterflies and the moths. As you can see here on the left side, they were made really beautiful pictures. You can also find all the records on iNaturalist in the gallery of Gernot Kunz. Among the butterflies, there were 56 species found, which is at least 29% of all known species in Bosnia-Herzegovina, and 222 species of moths. Vera? And now you see even more nice pictures. This is one of the 17 species that are new country records. Here is Diacrisia purpurata. It's one of the 2 species that are rediscovered after more than 100 years. And last but not least, there were 3 butterfly species that are on the list of protected species of the flora and fauna of the Republika Srpska, with this beautiful Apatura iris. And yes, that's fine. So thank you very much for listening.
Okay. Danke, Michaela. Thank you very much. And I'm going to continue with a few slides from the fish part of this whole science week. So I was part of a team investigating the fish species there, and we've been applying several methods to investigate the Neretva River. We applied the quantitative electrofishing method that you can see here, 2 colleagues of mine preparing to sample the river. Then we also sampled qualitatively, and we also took some DNA samples. We took samples of the fish, like tissue samples, and we also did some eDNA samples where you sample the water and you collect the DNA that is drifting in the water to do some analysis on that. Next slide, please. Here we can see some results of the upper and middle section that we sampled. There we found basically 4 species, bullhead species, stone loach, minnow, and brown trout. So what you see here is the length frequency diagrams. I just want to say a few words about the brown trout. Overall, we did find very well-developed populations of all species. Maybe for the brown trout, it's interesting to mention that we could see a very nice gradient of high numbers of juvenile fish in the upper sections and lower numbers in the lower sections of the rivers, which indicates that the river is, that the continuum is open, that the fish can migrate, and that the headwaters are used as nursery areas. So this underlines the pristine situation in this river basin. If we go on, then we talk a little bit. I talk a little bit about the qualitative data. We went snorkeling, which is another method that is applied in clear rivers, especially if the river gets too big or too deep to sample with the electric gear. Then we do qualitative assessments via snorkeling. So we did that in Klavadicevo, further downstream, and there it was very exciting to detect the soft-mouthed trout, Salmo-Optus irostris. It's an endemic species to the Balkany region. It's very specialized to those rivers there, and we were very happy to see this species there. We also did some interviews with the angling associations, and they told us that the soft-mouthed trout can still be found around Klavadicevo and also 25 kilometers further upstream, which is actually in the area where the construction site is now going on for the hydropower plant. They also told us that marble trout can still be found in the river, even further upstream than Ulog. So quite some interesting results there as well. Concerning the molecular analysis, so the analysis of the genetic samples, there's still some results pending, but what we know now already is for the minnows, so for the Foxinus, we detected a haplotype which was already described before. Some expected results, and for the bullhead, a very interesting result is that we found a novel haplotype. So a haplotype, or this haplotype, kind of is a marker for geographical uniqueness. So we were able to describe this new haplotype, and the bullhead that we investigated, their closest relatives are situated in the Po Basin, so very interesting here as well. And yeah, for brown trout and for the eDNA samples, we are still waiting for more results to come, and I'm happy to report about this in the near future. And that's it from the fish side. Thank you very much, and I want to hand on to Gabriel, probably.
Yes, you do. So among us, there were quite a few people who considered themselves as ecosystem ecologists, and these actually have a primary disadvantage. They usually can't identify any species of a special group, so they have to do complex graphs and ecological analysis. And these people were basically occupied with trying to find links between biodiversity and ecosystem functioning. And the primary idea behind this, and that's shown in the black triangle, is that an ecosystem function, and I'm talking about things like primary production, for instance, or decomposition of leaf litter, is connected to both the organisms, so the biota that live in a specific place, as well as the habitat features. So if that's true, then also biodiversity should have a link to several functions, and we like to talk about multifunctionality in this context, or functional diversity would be a different word for it, as well as to habitat diversity, meaning basically the heterogeneity or the diversity of habitats that are available in a specific ecosystem. So we looked into this by throwing 15 scientists and students on 3 different topics. And those were an analysis of food web structure and trophic diversity of macroinvertebrates. So some of them you have seen already. And then there were 2 more topics, but they're coming anyway in a second. So let's go through the first one. Here's a few results from food web structure and trophic diversity. And the group was led by Edurne Esteves, and she led a group of students into investigating the macroinvertebrate community's food web structure. So she was looking into basically the same animals as other people did, but never bothered about actually identifying a specific species. Rather, she tried with her group to identify the linkage to specific environmental variables, identify at the level of a relatively rough biodiversity assessment. And most importantly, the samples were also used for a relatively expensive and effort-time analysis to analyze a food web that actually involves mass spectrometry to identify a stable isotope composition of body mass. Let's have a look at how this works. Next slide, please. If you look at stable isotopes, you look at stable isotopes of basically body tissue, and organisms as well as us, we're made up of carbon and nitrogen and various other elements. But there's not only 12Z and there's not only 15N, there are stable isotopes as well. So our body has a very specific isotopic composition. And that's true for other organisms in the food web too. And the thing is, you normally get the isotopic signature, so to say, and that's what's shown here on these two graphs, delta 13C and delta 15N. You normally get the signature of the food you eat, right? Or at least closely, right? And by analyzing these two different isotopes, you can actually reconstruct a food web as shown in the very small graph in the center. So raw data, for instance, looks like the graph on the upper right. This is basically one of the raw data from one of the sites we looked at. The various colors are specific organisms from the macroinvertebrate community. The green dots stand for organisms that like to feed on algae. The brownish ones are more leaf eaters or detrital feeders, and the reddish ones are predators. And then what we can do with this raw data, this stable isotope data, we can actually compute things like trophic position or percentage allochthonous material that's used to build up this biomass. And the allochthonous material is a nice technical wording for the amount of leaves versus algae that actually support this food web. So let's have a look at what this data actually looks like. Next slide, please. This is the mountain of data that this group generated, and there's absolutely no way that I can go into detail here. But what we can do with this data is we can analyze questions or answer questions like which basal resources are used in a food web, for instance. How important are leaves compared to algae at a very specific place for the macroinvertebrate community? How long is the food chain? We can do so by computing things like average or maximum trophic position. We can also ask and answer questions like how complex or how diverse a food web is, or how balanced specific trophic links in this web are. So it's a very, it's quite a sophisticated type of analysis. Let's look at more integrative results. Next slide, please. What we found, I would say, is nicely summarized by the idea that yes, there's something like a river continuum if you follow the upper Neretva from the source down to Klavaticevo and even towards Konjic. But this natural river actually has a lot of discontinuities, and that's probably true for many real ecosystems. And it's in this sense, the Neretva is a rather special system given its size and the high landscape heterogeneity and diversity that it actually offers. So in terms of community composition, we actually find a little bit of continuity. So we see changes from headwaters to downstream sections, and you can see this in this graph on the left side. You can see the green circled sites, every letter here, every number here stands for a specific research site. They're quite similar, they're further upstream, and they're quite separated from 3 sites that are further downstream. But there also are a few places that are very special, like for instance, is number 1, which is a tributary far upstream. So you can see that there are there's something like continuity when you move downstream through a river network, but there's also very discontinuous places where things all of a sudden become quite special. And the maps give you a bit of an idea of what we can do with this data. You can see, for instance, as a color gradient, you can see measures like diversity. So this is a measure for biodiversity. You can see measures for mean trophic position. That's what the abbreviation MTP stands for. Where you can see how the percentage of allochthonous material or isotopic evenness, these are measures that we can derive from the stable isotopic assessment behave. And if you just look at the color gradients, you can actually see in some places there's a nice continuous development as you move along an increasing river size, and in some places there's very distinct discontinuities in this that actually show a relatively high functional diversity of the food web at these places. Next slide, please. The second group in this group of scientists that looked more into ecosystem functions actually looked more specifically into functional diversity and actually tried to focus on mostly microbial-driven functions, right? And these were specifically 5 functions targeting things like primary production, ecosystem metabolism, biofilm production. So this is the slime on the stones, basically, and what it produces in specific enzymatic activities that are related to ecosystem functions. And then there's also an experiment on organic matter decomposition. So what this group did basically was looking at all these functions at the various places. Next slide, please. And this is now maybe rather nerdy science, but what we can see is that we in general have rather oligotrophic headwaters, and when we move downstream, we have more influence and more nutrient input from terrestrial vegetation and human land use. This is a very classic pattern for a river continuum. And what we achieved in this group was basically showing how 5 different ecosystem functions react to these changes in nutrient nutrient content and increase in river size, basically. If you look at the map, you can see a color gradient from red to blue, and basically this color gradient shows a measure for specific ecosystem functions, these 5 different functions, and the more similar the color of 2 different dots are, the more similar these 2 different places would be assessed based on these 5 functions. We can very nicely see that there's sort of a gradient towards Uluk, and then there's a bit of a restart, right? So at Uluk, you can see there's a switch from a blue dot again to a red dot. So here, the river continuum experiences kind of a reset. And that's pretty interesting because this is the place, this is the region where the current hydropower construction site at Uluk is actually happening. And this is, from this analysis, basically the place where the functional diversity from the landscape would be highest in our analysis. So Uluk presents itself, basically the area around the construction site, as a hotspot of functional diversity in this place. You can also look at this in terms of a multifunctionality measure. Now, this is maybe really nerdy ecological analysis already. Basically, this tries to look at all 5 functions at once and sees whether there's any trend. And yes, there is. Again, we can see that there's a nice shift from upstream to downstream, but close to Uluk, there's quite a bit of heterogeneity. The bigger the river is, but also specifically ammonium input actually increases functioning quite a bit. Beyond this, and that's again shown on the right side, we should look beyond average functioning and actually also realize that there are specific tributaries that actually have very specialized functioning in the landscape, right? So it's as very often, it's not just about the big river and how it behaves, it's also about specific tributaries in the river network. Next slide, please. One of the functions, so to say, that the third group was looking at is pretty specific, specifically in a hydropower context, and that's a group led by Martin Dalvay, who actually looked into greenhouse gases. And these people actually partly traveled by boat, partly they hiked the river continuum to measure greenhouse gases, specifically methane and carbon dioxide concentrations. And the idea was to identify the importance of very discontinuous inputs by karst springs, for instance, but also by heterogeneity of the river channel, as I have already pointed out before. The group characterized hydromorphology to compute things like gas exchange, and I've already introduced you to the map they produced before. And then they measured discrete gas samples wherever this was possible, from a raft, from a backpack, or from grab samples that were partly collected over 24 hours. Next slide, please. Here's basically 3 different maps that show the effort that this group has done. The first one is the one that I presented already. So this gives an impression of the physical diversity, the physical heterogeneity of the system. This is a measure for gas evasion velocity. So it's a measure for how efficient the water body exchanges gases with the atmosphere. And then the 2 others actually show maps of concentration of carbon dioxide and methane. And here I would again like to point out that there's quite a variability as you move through the network. And importantly, the hotspots of carbon dioxide concentration are not at the same place as those of methane concentration. Next slide, please. If you bring these variables together, you can compute something like a gas emission. So this is basically an emission of a greenhouse gas flux or a greenhouse gas like carbon dioxide or methane. Again, I would only like to point out the large heterogeneity. So there's quite a bit of variation when you walk through the river from one place to the other. And this is because there's different concentration. It changes dramatically over only a few hundred meters, but there's also different physics in the channel that actually drive this. The hotspots of carbon dioxide and methane emission don't align, and that's in fact the consequence of the functional diversity that we have in the system. So you can't or maybe you can actually look at some of the numbers. I would only like to point out that these greenhouse gas emissions are, of course, a natural feature of an ecosystem. The emissions that we measured are very low relative to other headwaters in the region or in the world, and they are for sure magnitudes lower than what would happen if a reservoir is built. So around Ulok, we can expect if that reservoir is filled, we probably have to deal with much higher greenhouse gas fluxes because of this change from a running river to a standing water reservoir, basically. With this, I'm moving on to and pass on the torch to where is Spela somewhere.
Hello, everyone. Do you hear me? Yeah, cool. Hi, I'm Spela, and I will present you the work of the subterranean team. So we are back to diversity of the Neretva, this time underground. So first of all, I want to tell you something about subterranean habitats. Probably when you hear the word subterranean, you think about caves in limestone in karst areas. And in fact, half of Bosnia and Herzegovina is karst area with numerous caves, sinking rivers, and karst polyas. But less known is that important subterranean habitat is also under the river. And this is a so-called hyporheic or interstitial between the gravel. So these tiny spaces filled with water under the river. And the river communicates through this hyporheic with groundwater. And we all know why groundwater is important. Basically, a lot of countries drink it. So, and there are animals in these permanently dark places. And these subterranean animals are quite special, not only because I study them, but also because they're adapted to permanently dark and quite harsh environment. And moreover, usually they're highly endemic. Some of the species are found only in one specific spring, in one specific point on the river. So what we did, how you approach these groundwater habitats? Well, you use a brute force. Our work included a lot of hammering and pumping and filtering, because basically we wanted to get to the water that is under 1 meter of gravel. And after we got this water, we filtered it, we checked for the animals, and the majority of work actually followed in the lab. All the sorting of the material and determination, and also DNA analysis of interesting groups. I will just summarize quickly what we found. We sampled 6 different gravel bars, 6 different springs, and we went into one cave. And in these gravel bars, the diversity of samples actually quite surprised us. And I have to tell you here that various taxonomists helped with determination, and not work is done yet. We specifically focus on subterranean groups, but because this is intermediate habitat, you can get also surface groups. So under the rivers, we found more than 30 species, and at least 10 of them were obligate groundwater species, which means that they cannot survive in surface waters. They can survive only in groundwater. And wait for this, at least 5 of them are probably, or most certainly, new for science. And some other, and we will probably find some other interesting findings in the groups that we did not determine yet. In springs, the diversity was even higher. More than 50 taxa was found there, and 7 of them were obligate groundwater taxa. And 2 of them were the same as in gravel bars, new to science. Visit of a cave was quite a challenge on its own due to quite demanding approach, and the fact that we didn't even have a location of the cave. But we managed with quite a lot of help of locals, and we actually quite thoroughly sampled one cave where we didn't find water, but we sampled terrestrial species, and we caught 8 obligate subterranean species. And now I would like to just quickly present the interesting findings, the new stuff with molecular methods. So with DNA comparison, we determined new species of amphipod from the genus Niphargus for science, and probably 2 new species of isopods. And with morphological examination, we found 1 new copepod and 1 new ostracod. So a lot of work is still needed to be done here. And here I can tell that we found, I think, 5 specimens of Niphargus. So it's, well, we have to go to next Science Week. And in second part, I want to present part of the results of the bed group, because we focus also on this interesting group. You probably all know that beds are quite important elements of river and forest ecosystems. So what we did, we used mist netting and we recorded their voices with bed detectors, and we checked the buildings for their roosts. And we found 15 species, which is half of all known species of bats for Bosnia and Herzegovina. And 3 of these 15 species are on Annex 2 of Habitats Directive, which is actually important because this makes this Neretva area a candidate area for Natura 2000, especially because we also found nursery colonies of one of the species. And on this quite nice photo, you can see one of the important findings, the greater mouse-eared bat. And the other 2 were horseshoes bat, lesser and greater horseshoes bat. And you can see on the photo on the left that we also found a nursery colony that lives in one abandoned building in Ulov. The last interesting thing was finding of Akatoe bat, because this one is specifically connected with riparian forests and was only a second record for Bosnia and Herzegovina, and first time confirmed also with DNA analysis. So the Neretva is actually quite diverse area in terms of subterranean diversity and also bed diversity. So a lot of work is still needed to be done there. And this would be it from my side. And I think that now, okay.
I'm coming back to just one slide. The results that you have seen today were in partly, at a very preliminary stage, already published last year at the end of June, when we managed to pull together results only about 4 weeks after we came back home in a preliminary report. And we plan to actually have a publication, basically a scientific book in the proceedings of the Natura Slovenia. And I'm very happy to actually say a big thank you to all the scientists that have already submitted a contribution. So there are several papers on our desks at the moment which will be published very soon for scientific peers. And on top of this, we hope that we can also publish a final report. And just from the picture that you can see here, we hope that this is going to be readable for a much larger audience and not just for the scientific peers. And with this, I'm passing on to Vlado, I think. Yes.
Yes, thank you, Gabriel. I hope so that all of you hear me, so I will try to continue. I'm Vladimir Topic, Center for Environment. I want to say something about the impacts of this Neretva Science Week from the last year for our country and everything. So first of all, I want to say that the data for this region before the Science Week, we have some data actually, but not even this this very rich data as now. So when we talk to people before the Science Week, we talk that we will organize this event. We do some researching before, and a couple years before we do this researching for some biodiversity group and everything, but this some big event like this is not being present in our country. So we try to create with this scientific work in this region and to collect more and more data, because that is really important for our country, because we will have some results at the end for our protection that area and the protection of that river, as Neretva is very rich and on a high level of biodiversity. So for first of all, the impacts is incredible, because all the locals, local people, all the people from other local groups as NGOs or institutions have realized that we succeed to do this Science Week on one river with as a camp for 7 days and everything. And after a few months we have this preliminary report, and even now we have a great data. And the media start for now they have more and more interest for this area, and they want to know what is this, what we have actually in this area, and what we do next. And so in that point of view, we really need this data, and we need to continue to collect all this data for that area. Also, it's important that some international news and some international companies and news create a report for this this Science Week, like National Geographic. So thank you, Nick, for that. The people are be present in this area and try to figure what's happening in that area actually. And after all, we success to talk with people and to spread the word actually about this area and about the Neretva, because from the beginning we know that all that area is, as I said before, is on a high level of biodiversity. So now we are pretty sure for that. And the important thing is that we all of this data we collect and put in protection study for the upper Neretva. This is upper part of the Neretva River actually, because all this stretch on the picture is planned for the protection. We start with that initiative, and we hope so that definitely we collect. After Science Week, we collect all this data and put in the protection study. Now the protection study is on the table in our institutions, and we will try to continue to push them to do something for the nature and the people actually. Biggest impact is actually that we use all this data from the Science Week and for the case for Bern Convention case actually, because we before a few years actually we start the case and do the complaint on the Bern committee of the Bern Convention. And in October last year we have a on the spot appraisal in that area, and that is the period when we have all these informations from the Science Week, and that is actually a good time for that meeting and also for negotiation on this Neretva case. And Bern committee for the Bern Convention actually, standing committee for the Bern Convention actually opened the case in the November in the beginning of December and last year, because they realized that this is a really good, a really important area, and that we cannot allow our government to continue with this, not only Ulog project, like Ulog Dam is, even we need to stop other projects on the Neretva and on the tributaries of Neretva. So at the end, we collect many data for this area, and we use that data to negotiate with other group or with institutions, and this is I can say that this is a good way for us to do something better for all that area, like Neretva River and all the Neretva basin. Thank you. Vera, probably you can continue, and Uli can speak after me.
Exactly. Uli will jump right in here.
Okay. Yeah, hi everyone. It's a pleasure for me to reach out to the next Science Week. So to say a few words about last year, I think the ones who joined us in that week would agree that this was not only because of assessing data, it was one of a great week, one of my favorite weeks in the whole of last year. And we decided internally we will do it again, the Science Week, again on the Neretva, and you see the dates here, May 30th to June 7th, so a bit earlier than we had it last year. You can arrive a bit earlier, and the camp will be in Ulog, again, the base camp, but I will show you what is different. Next slide, please. The Neretva is more than just the upper part of the Neretva. The Neretva is per se one of the best rivers we have left in Europe as a whole. When I say the Neretva, I always mean the whole Neretva system. So not only the single river from source to mouth, but also the tributaries. We have basically decided to have our assessment this year on 3 locations. You see this is the upper segment where we have been last year. We will do things differently here. And then there is the Ljuta River. That's the second place we would like to do assessment. And the big circle is connected to the downstream part of the Neretva, but it's close to the upstream part. It's the karstic area where a big hydro scheme is under construction for decades now, the so-called Upper Horizon, where they try to divert the groundwater away from the region and direct it into the Adriatic Sea via hydropower plants. But it sounds maybe complicated. This is just an overview. Next slide, please. Yeah, so this is a picture you've seen before. That's the Upper Neretva. And what is so amazing about this is not only that the river is great, but you see the slopes, it's covered with forest. It's completely covered. You hardly see any, I don't know, forest trails or roads. It's a roadless area, which is absolutely rare or almost unique within Europe. So this year we will do transects, not only in the river, but we will assess all these different types of species, whether they're insects or mammals. Also in the mountains, you see the red dots here. These are what is symbolic, no? But it's transect. So we go with our assessment from the one peak down to the river, up again to the other peak. Because the main question that we try to solve or come close is, what is a river? What's a river? Everything is connected. Pantare, that is also true for the sites. And the river has an effect on the forest, but the forest has an impact and effect on the river. And this is what we're trying to do. So same location, but different tasks, going to the forest and get new results. The second one is the Ljuta River. That will be not for the masses, because it's a narrow river, and 8 dams, or in some scenarios, even 10 hydropower plants are being projected in this river. It's all diversion. Contrary, it's like in the upper part, so they would divert the water away. It's moving forward these projects, and we need arguments to battle these projects with our lawyers. So it's not only we need the public pressure that people understand that the Luta is important, but we have a legal team in the Blue Heart campaign, and they need arguments. And so this is important to assess. Next one. Yeah, that's a bit the way the Luta looks like. The third place of the Neretva Science Week is the so-called, it's difficult to pronounce for non-Balkanese, Nevesinsko polje. These poljes are, wait, you stay. These poljes, no, back please. That's too quick. Can I have one slide back?
Yeah, I'm trying to go back, but I'm going forward.
Okay.
I'm sorry.
Okay. Now these
Oh,
poljes,
there we are. There we are.
yeah, these poljes are unique ecosystem in the karst that exists nowhere else in the world than on the Balkans, mainly in Bosnia and in Croatia. They have karstic springs, so these areas, these meadows are flooded in springtime, and then the water gets back into the ground. So next slide, please. And since the 1960s, I think, under when it all was Yugoslavia, there was this plan, so-called Upper Horizon. They want to divert the water away from these poljes. This is, I think, Fatačko polje. It's another polje. And you see what they plan to do. They want to build long concrete canals and then tunnels through the mountains. So it's like they build subways, like U-Bahn, which are partly on the surface and partly underground. I think the total amount is 150 kilometers of hydropower infrastructure that they want to build there. And the Nevesinsko polje, where we are, is the most crucial area of this whole project, and probably the most crucial area, next slide please, for the whole Neretva basin. One of the rivers in this Nevesinsko polje is Zalomka. I can explain that was in April 2 years ago when we did a fact-finding mission for the Neretva Science Week. This water is flowing into a sinkhole, but there is fish species, for example, that live most of the period of the year in the darkness, in the underground. And they, with the upcoming water in spring, they migrate into these little rivers, spawn on the grassland, and then go back through the sinkholes into the karstic cave system. That's amazing. And by building these Upper Horizon projects, at least 2 fish species would go extinct if we allow that to happen. That is actually the next one, please. This follow the Zalomka river. The next slide. This is the, oops, that was too fast. Yeah, that is the end of the Zalomka river. That is the famous, they call it Sinko, very to the right, where you see like the cliffs. That is where the Zalomka flows in, and it can have 21,000 liters per second can go into that cave system. The enormous interesting thing is that this whole system is connected to the other side of the mountains, to bigger tributaries to the Neretva called Buna, Bunica, and Bregava. They are connected. And the goal of the hydropower plant is to build exactly here a big dam to stop the water to going into the karstic system. The impact of that is hardly known, but what is for sure clear is that the rivers on the other side will have less water. Species will go extinct. So there's a lot more to tell. It's complex. It's really weird. I also myself never encountered such a way of hydropower schemes. And we want to go there with all the team during the Science Week and assess data to also collect data for our legal experts to battle this project. Next, is there a next one? I don't know. Yeah, thanks. That was from my side. So next year, this year, end of May to 7th of June, another expeditions type. We will have the camp in Ulok to summarize it up. That will be the base camp. And from there, we go to Upper Neretva, Luta, and the Nevesinsko polje. 3 different types of wonderful and beautiful and enormously diverse and largely unknown areas. Thank you.
Thank you, Uli. I'll just quickly jump in for the explanation of this slide. Namely, there were 3 missions already explained by Uli. The idea is like if there's somebody else that has a whole different idea and that has interest in the Neretva and has a reason to say like, okay, my mission might contribute to fighting the construction of dams on this river, so stopping this this kind of destruction of the ecosystems, right? We're open to other ideas, right? So that could be a 4th, maybe even a 5th mission. But I'll give it to Spela now to explain how you can potentially join the next Neretva Science Week.
Hi everyone again. So basically, I will communicate with you. Probably if you will join the next Science Week, I will be the one who will tell you where to go and how to come there. Please contact us, write us an email, write us any ideas that you have. If you want to contribute to the missions that Uli explained, or you have a completely new mission, even if it sounds crazy, please tell us. Join our network of scientists for Balkan rivers. You can join through the webpage that is on this slide. You can also find all the information on the webpage, and also we have Slack for communication, and I posted the link in webinar chat. Some of you already responded. I'm really happy for this. So I'm hoping that I will see you on Neretva at the end of May. Thank you all.
Great, thank you, Spela. Thank you everyone for joining us today. There would be time for some people if you actually have a question, if you want to dive in a bit more. We would have a few more minutes. Otherwise, I'll take it as it was a very clear session and everything came across well. In that case, I would especially also like to thank all the scientists that joined today, that were behind all those results, and more is coming soon. Watch out for the actual report coming out, which will be published. Thank you, Uli and Riverwatch and the whole Save the Blue Heart campaign. Thank you, Annika, as well from Euronatur, for helping with this webinar, but also with the whole mission of last year. Thanks, Gabriel, specifically for hurdling all the scientists all the time. And thank you everyone that joined us today to learn about the results, and hopefully we meet more of you in the coming year at the Neretva. I'm unfortunately not going to be there, but you're in good hands with Spela. Vladimir, good you opened your video, because a lot of thank you to you and your team as well, from the Center of Environment, because thanks to you all the logistics were possible, and good luck with the next one, because the plans are a bit more crazy, I would say.
Yeah, I would like also say thank you to you, Vera, and special. You know, it's kind of your last activity, you know, within this frame of the scientists for Balkan rivers. It was a pleasure of having you. I envy you sometimes seeing you where you are. You know, you look like to be on holidays in Peru, but you are not, I know. But it was a pleasure and a great achievement to develop this whole network. Not only the Science Week in principle, but there is this for everyone who is interested. There's a scientist for Balkan rivers. Go to the website balkanrivers.net. You can click on that. You can participate in the science community for the whole Balkan rivers segment. But that was you developed it. You created it with us together. And thank you very much. It was great. Already miss you.
Very, very welcome, and thanks to you all, because without you all, it wouldn't have been great. It was really cool working together with this team. And I sometimes regret my decision already, but it's good. Cool. Well, I think that's the moment to close this webinar now. It is recorded. I should have mentioned this at the very start. I did not. So in case you didn't want to be there, sorry, it's going to be published online as well. So for anyone that wants to look back, it will be online, and you can pass it through to other people as well.
Yeah. Hey, there's no way we can show the video, no?
Well, I can put it up again, but it was just like we tried it and it was super shocking. Like super bad
Or just
qualities.
Or just you can send the link in the chat.
Yeah,
I think that's fine.
At
I think
least.
that's the best.
It's already-
We sent the link so we can all just spend 5 minutes watching the video.
Yeah. Yeah, it brings across the
3, 2,
vibes
1, we'll
so well.
together going through.
Hey, Vera, did you mention when the final report will be available, or somebody, or Gabriel, did you say that?
Well, I didn't make any promises. No, I for all the scientists that are here, I'm really happy about all the contributions that are currently on my and Maya Zagmeister's desk. We're making sure that the contributions go through a peer review process before you're probably asked for a revision, and then eventually we'll hope to publish it by maybe a reasonable estimate. This is going to be April. And alongside with this, I think we should follow up the idea to also publish a more readable, more digestible version similar to our preliminary report. And I'm very happy about offers that come in from Katie Ellis, for instance, or forgot the name, I'm sorry, who offered to help us write like a more, say, accessible piece of text.
Great. I put the, once again, I think Spela did it already, but I do the link where you can see the video for the scientific. It's like a 3-minute video that summarizes the last event and, you know, transfers also a bit the emotions. Okay. Well,
We're looking forward for this year's Science Week. If you want to join a mission, yes, they are maybe slightly crazy, but that's the point. Or if you have another mission that's exciting to offer to us, then please just get in contact with us. Thank you, everybody.
Yeah.
Thank you.
Thank you, and
Thank you.
Thank you.
bye.
Thanks, everybody. Bye-bye.
Bye-bye.